fasting pathway. So what happens, after let's say a person is not eating, right? So what is our objective in fasting? So what is our objective in fasting? Objective in fasting is, let's say we go back to the blood vessel again, right? Like same here, we went to blood vessels, has 100 grams, and it just increases way much more in feeding. Now we go back to same example. Objective is here. So normally, what is your fasting levels in of glucose?
I give or take 100.
100, right? Give or take. So 100, let's say. So why do you even need to maintain 100 grams of glucose all the time? Like at least 80 to 100 or whatever you want to call it, right? Why do you want to always maintain that is a question, right? 100 grams, right? So the reason you need to maintain that all the time is because you so let me just how can I make that simple is, let's say so I'm gonna just draw some rules here. So write down some rules, right? Like some, not rules, just a normal science. All cells in our body need what? ATP, right?
Mm-hmm.
So how do you make ATP? Well, through metabolic pathways, right? That's one, right? So that's number one. What is number two? So in order to make ATP, you can use either glucose or ketones to make ATP, right? Right? Number three rule. All cells prefer glucose over ketones.
Absolutely.
Right? And number four rule, sorry. There's so many rules, man. So all cells prefer glucose over ketones. And what else? Only cells that can use ketones must have mitochondria. Make sense?
Mm-hmm.
So now let me ask you, like, which cell do not have mitochondria?
Red blood cells.
Exactly. Which cells don't have mitochondria? Is RBCs, right? Mature RBCs, right?
Mm-hmm.
So which means if there's a RBC here, sitting here, right, in the blood vessels, and you don't have a glucose here. Let's say your glucose dropping down to, like, let's say, I don't know, it's like normal is 80 to 100, right? So let's say your glucose drops down to like 20s and 10s, right? So what happens is these RBCs, right, they won't have glucose, right? So now I'm gonna just zoom it in, this thing right here. I'm gonna zoom it in here. So this is the RBC here, right?
Mm-hmm.
So what happens is once this RBC is getting glucose, right, it doesn't have a mitochondria, right? So let me just see. Here, and here. So does it have mitochondria?
No.
So it don't have mitochondria. So it cannot do Krebs cycle, cannot do all the other stuff, right? So through the Krebs cycle, right, this is electron transport chain, right, in other cells, right? So you can make a lot more ATP, but it doesn't have mitochondria, right? So it cannot use that. So glucose goes to what? Pyruvate, right, in these cells. So these cells, RBC, can only do, so RBC can only do glycolysis to make what? To make ATP, right?
Mm-hmm.
So how much net ATP will make through glycolysis? Only 2, right? So why do you need to make ATP, right? Because, like, let's say there's a pump on the cell membrane of of the RBC, And then again, you have a sodium potassium pump, right? Remember the sodium potassium pump?
Yeah.
So sodium potassium pump here. What do you need for a sodium potassium pump to work?
ATP.
ATP, right? So this doesn't have mitochondria, so they can only have 2 net ATP that only come from glycolysis. So you need to have this glucose. So even if there's a ketones present in the plasma, it cannot use ketones. Why? Because it don't have mitochondria, right? So what happens is, you need to have this glucose to have this, ATP, right? If this ATP will, keep this sodium potassium pump working, right? Because you need to pump the K in and pump the sodium out, right? Remember the mnemonic?
Yeah.
Pump K in, pumpkin, right?
Pumpkin, yeah. It's two in, yeah?
Yeah, so
Three out.
so yeah. So pump the K in, okay? So at any time, if you don't have glucose, your RBC is not going to work, right? Which means you will not be able to maintain this, sodium potassium pump, which means you won't have energy, and this RBC can actually theoretically can undergo, if you don't maintain your membranes, these gradients, they can undergo hemolysis, right?
Breaking of the red blood
Yeah, breaking
cell.
down, right? RBC will hemolyze, and then again, what does your RBC do? Well, they carry a lot of oxygen, right? Because they have a hemoglobin, they can bind all that, oxygen, and then they just keep on, Otherwise, you will feel fatigue and all of that stuff. So for main reason, you need to maintain fasting glucose is for RBC, really, right? Like every other cells, your neurons, your cardiac muscle, your skeletal muscles, every cell in your body can use ketones because they have mitochondria, right? But they prefer, it's the same thing, right? Like if somebody gives you, like, I don't know, if you go to some restaurant, right? And then you get like a pasta, right?
Yeah.
Versus like some bread, like without anything. Which one you prefer? Like if you're hungry, you definitely will eat bread, right?
Yeah, of course.
But you really prefer like a nice cooked pasta, right?
Yeah.
If you go to cheesecake.
Of course,
especially,
of course.
right? So that's how it is here too, right? So, but the thing is, the absolute glucose you need at least for RBC, because if you don't have glucose, the RBC is not going to work. They're going to hemolyze, and they're not going to be able to carry oxygen and deliver to your tissues. They actually have mitochondria, right? And then you won't be able to make energy in those cells. So for that reason, so your objective is to maintain glucose levels is mostly for maintaining RBCs, right? That's really important because a lot of people don't, think that, when they are thinking of fasting, like, why do we maintain 100 grams of, like, for example, you go into the hospital in the morning, the first thing we see is, oh, wow, this patient has a glucose level of 20. Oh my God, just give them D50, right?
Yeah.
So why do we give D50 right away? It's because we want to make sure that, RBC are not deprived.
Absolutely.
Right,
Yeah.
so like we just start giving them a lot more, because every other cells can use ketones, right, but except RBCs, right? So that's why it's really, really important we maintain these sugar levels, right? So that is objective. So now the question is, how do you do? So fasting pathways. So our objective is to maintain 80 to 100 grams, and then these cutoffs always changes with the guidelines. Changes every single year. So 100 grams of glucose, right, in plasma. And for which cells mostly? RBCs. Yes, other cells love glucose, but the goal is to really save the RBCs from not working, right? Because these cells can only use glucose to make what?
ATP.
ATP. Right? And that's really important, right? So that's one. Okay. So now what are the pathways? What are the ways we maintain these things, right? It's mostly like 3 or 4 ways you maintain these, right? So blood vessel here, right? And there's 100 grams of glucose, right? You're maintaining it. So how do you maintain it? So I'll draw this little chart here, which will hopefully be more understandable. So let's say, let me see here. Let's say this is like somebody just ate like 2 hours ago. I don't say 2 hours ago, maybe like, I don't know, 1 hour ago. They just ate. And now, so this is 1 hour ago, so this is -1, right? This is 0 hours. So at now, let's say, after 1 hour, they ate, right? So the body is now, technically after 30 to 40 minutes, depending on how much you ate, again. So your body is now starting fasting here. So they were in fat state all this time, right?
Yeah.
Fat state. And now your body kind of shifted in. Right? So let me just change this. Into fasting state now, right? So now their body is into fasting state. So the question is now, what happens? So this is at 0 hours. So here you need to maintain your blood glucose. So now you're not eating, right? So there's no exogenous source. You're not eating, right? So all this glucose that is, now this is coming from your cells, right? Depending on what cells you're talking about, right? So your body is maintaining this glucose from endogenous sources, right? Because when you're fasting, you're not eating. So how do you maintain that? So like, let's say this is a, right here, this is like, I don't know, give or take 100 to 80 to 100 grams of glucose levels right here, this line, right? So you need to maintain these glucose levels like roughly around the same range, right? Because if you don't, your RBC will pretty much will not work, right? So what happens is, the first thing you can do is, easiest thing, so during your feeding pathways, right, you stored a lot of glucose, right, in the form of glycogen, right? So now the easiest thing you can do is you can take the glycogen and lysis them, right? So this is what we call glycogen-lysis, right? So this glycogen, and where this glycogen is coming from, remember we said this glycogen is mostly stored in where? Liver and skeletal muscle. So it's really important to pay attention here is, this glycogen right here is coming from skeletal muscle or liver? It's mostly coming from liver, not your skeletal muscle.
In the first, in the time frame that you're, okay.
So skeletal muscle, because again, which hormone is elevated in fasting pathways? Is glucagon, right? So now you have more glucagon than insulin. You see what I'm saying? So now what happens is, if the glucagon is higher, your liver has a receptor for glucagon. So you can break down, you will start breaking down glycogen. But your skeletal muscle don't have a receptor. For glucagon. Understand?
Okay.
So which means during fasting stage, the glycogen that is you're breaking it down is only coming from the liver.
Gotcha.
It never comes from your skeletal muscles.
Okay.
So then we will talk about like what actually the glycogen that you stored in the skeletal muscles, what happens to that. We'll talk about that later.
Okay.
But during fasting pathways, fasting stage, the amount of glycogen that you're breaking down, it's coming from the liver, not the skeletal muscles. Okay, because skeletal muscles do not have what? Receptors for glucagon. So you cannot activate glycogen breakdown in the skeletal muscle if you don't have the receptor for glucagon.
Okay.
Right, so you only have liver has the only is organ that actually have the glucagon receptors, right? So whatever the glycogen is stored there, it's going to start breaking it down, right? So what happens is up to 2 to 3 hours. So your glycogen, let's say this is your glycogen store. we'll just pick some different color here. I don't know. So this is, let's say this is a glycogen store right here, right? So that's, I don't know, this some glycogen store. Now it's going down, right? So now you used up all your glycogen, right? Stores. So you had some glycogen stores here, right? Something, I don't know what, I don't know, like, maybe some grams or whatever, stored in the liver, right? And now you're breaking it down, you're breaking it down, you're breaking it down. You used up all your glycogen, right?
How long does that take on average usually?
Usually 2 to 3 hours.
Okay.
It depends, like if you're working out and everything, if you need more glucose, right? If you also not eating and you're fasting, you're running. So what happens? Your RBC needs to work harder. Your cells need to work harder. They need to make more energy, and you're not supplying exogenous source, right?
Right.
So your body actually needs to make more glucose to keep your body running, right? So for that reason, you're going to start breaking down this glycogen even faster.
Mm-hmm.
Normally, if you're not working out, if you're sitting down, for example, for the most part, we're doing notes in the hospital, right?
Yeah.
So you're literally burning down your glucose, right? So that's what happens here. You understand? So let's say you used up all your glycogen stores, right? And then now you're still maintaining, right? So your whole purpose was to do what? Your whole purpose was to maintain this glucose levels, right? In this range, right? Up and down a little bit. And this, right? So now, once you run out of your glycogen, you already run out of this, right? So what is the next thing you'll do? Well, you need to start making gluconeogenesis. So now you need to make glucose. Mm-hmm. Neo means new. Genesis means synthesis, right? Which means you're making glucose from scratch. Make sense?
Mm-hmm.
So anything you're making from scratch is going to require a lot of energy, right? So now you burn down your glycogen stores. Now you need to make from gluconeogenesis from the scratch, which we'll talk about now. So what happens in gluconeogenesis? Let's talk about gluconeogenesis right here, right? So what happens is there's a pyruvate. So remember, when we had a glucose, now we need to get pyruvate from sources, which we'll talk about, and now you need to do what? Now you need to go backward, right? Before we were burning down glucose, right?
Mm-hmm.
And now what are we doing? We actually need to make glucose. Now we need to go instead of going this way, now we need to go actually backward, right? Because you had some glycogen here, right? You already shifted towards this way, right? And that's gone in 2-3 hours.
Yeah.
So now, if this is not there, you need to start making it from scratch. Because if you don't, what will happen to your this levels right here?
It'll start dropping. It'll tank.
It will tank. So what happens to your RBCs here?
They'll lyse.
They will lyse and they will not work, and the person will die, right? That's why they will have mental status and everything. We start injecting them D5, right, to maintain their glucose levels. So that's why it's so important to make sure, right? So now this gluconeogenesis, mostly right here, it's mostly like it's maintained by liver. Is the most important organ to maintain that. Okay. So, what is the clinical correlation? If the person have hepatic liver failure, hepatic liver injury, and if they're having hypoglycemia, which means after they used up their glycogen stores, right? So this process is mostly gluconeogenesis is maintained by liver. And if you have a liver injury, liver failure, will you be able to do this?
It would be very hard.
Right? So which means in liver failure patients, what is the first lab we check? We check for hypoglycemia. If the person is keep on having hypoglycemia, we check their LFTs, right? We make sure they don't have a liver failure going on, right? So that's the most important thing, right? So here you need to maintain their glucose level. So what happens is as you're narrowing down this, glycogen pathways, there's some level of, gluconeogenesis starting, right? So as this going up, as this glycogen store going down, this gluconeogenesis is going up and up, increasing in activity, right?
Mm-hmm.
It's increasing in activity, and then it levels down. And now you're maintaining your, glucose levels, right? Like that.
Through a completely separate
Right.
pathway.
It's a separate path. That's completely, yes, completely. So now the question is, so in order to do gluconeogenesis, which is this process right here, right? What do you need for that? In order to do gluconeogenesis, you need your substrate. What is the substrates usually? Substrates are mostly like, pyruvate is the most important one. And you have like some oxaloacetate, but most important is pyruvate. So this pyruvate gets converted into oxaloacetate, and then eventually PEP, and then eventually we'll go to, glucose. We don't really need to get into nitty-gritty details of that. The most important thing is, where do you get your pyruvate from? That is the most important stuff, right? Because if you need, that's your raw material. That's your substrate. So where do you get that from? So your substrate, what is the source for that? So substrate, source is your proteins breakdown. Okay. So these protein breakdowns, remember like in first semester of med school, we talked about like glucogenic amino acids, ketogenic amino acids.
Right.
Remember that?
Yeah.
So this is called glucogenic amino acids again, right? Amino acids when you combine the amino acids, they will make proteins, right? Mm-hmm. So you break down the proteins. So proteins broken down into amino acids, and amino acids are two kinds, either ketogenic, which will give you ketones, and there's our glucogenics, which will give these substrates right here, right?
Oh.
Right. So there are some amino acids that actually are both. They give you ketogenic and glucogenic, but the most important thing is we really cared about is mostly glucogenic, right? Because glucogenic amino acids, when they're broken down, they will give you pyruvates, right? A lot of pyruvates, and then some will give you oxaloacetate too. But this will be converted back to here into glucose, right? So that is one thing you need, really important. What is the next thing you need? Well, there's a first enzyme that is going to take the pyruvate and convert it into oxaloacetate. There's something called pyruvate kinase. Okay. That is the most important enzyme. That is upregulated by glucagon. But most importantly, acetyl-CoA. Most importantly, acetyl-CoA. Very, very important. This very important. If you do not have acetyl-CoA during your fasting state, you will not be able to convert pyruvate to oxaloacetate, you will not be able to make glucose.
Okay.
So what happens? You will become hypoglycemic in fasting state, right? And your RBC will not work, right? So what happens is, while you're doing this so that's number one, because remember, when you actually went from glucose to pyruvate, remember when we break down the glucose, we made 2 net ATP, right?
Yeah.
So you need to put some of these ATPs back now, right? Because if you made ATP while you burned down the glucose, in order to go back, you need to put these ATPs back.
Is it a lot more? Is it the...
That's still, like, you still need 2 net ATPs putting back.
Oh, wow.
Right?
Okay.
So that's your, so this is your number second requirement. Number third is this requirement, right? So you need ATP to put back to drive the process backward. You need acetyl-CoA to actually make the pyruvate kinase working. And you need the substrate to begin with, right? So again, 3 things, right? So for gluconeogenesis, you need 3 things. So what is the one thing is? Substrate, which come from where?
Amino acids.
Protein breakdown, right? Same thing, amino acids, right?
Yeah.
And what is the next thing you need? Need the enzyme working, which is pyruvate kinase, right? What is a third thing? ATP, right?
Mm-hmm.
And fourth thing is acetyl-CoA. Acetyl-CoA, why? Because you need to make pyruvate kinase to work. If you don't have this will not work. This upregulates the pyruvate kinase.
Yeah.
So now let's talk about the sources, right? So you have your so where does your protein breakdown come from?
Your muscles.
Exactly. Your which muscles first? Mostly skeletal muscle, right? So what actually happens when you start running out of your glycogen, you kind of start breaking your skeletal muscle, and once it's like completely out, this thing is even speeding up, right? The skeletal muscle breakdown is speeding up.
It ramps up.
Right?
Okay, yeah.
So that's why if you're not eating more than 3 or 4 hours to maintain your fasting glucose levels, right? You're breaking down your own skeletal muscle. That's why, like, if the person is not eating for 8 hours, 9 hours, they're losing a lot of their muscles too. And that's why bodybuilders, what they do, before their body shifts into gluconeogenesis, they just start eating right after 2-3 hours, right?
Wow.
They fill up their glycogen stores already again.
Wow.
And then they don't let their body go into gluconeogenesis.
Wow. So they're just trying to circumvent them breaking down their own muscles.
Exactly.
Yeah.
So that's why, like, hence, they say how many meals they eat. They have to eat like every 2 to 3 hours, right?
Yeah, they say it's like a chore. Yeah.
Right. That's why they do it, because like they may not know this, but they have been told. But this is the chemistry behind it. Is that they don't let their body go into gluconeogenesis, because again, for gluconeogenesis after 3 hours you need 4 things, right? Protein breakdown, which has to come from the skeletal muscle. Well, you worked so hard for it, you don't have to break it down, right? And the second thing is what? You need pyruvate kinase. And pyruvate kinase come from, who activates that? Acetyl-CoA, right? The most important thing is ATP, right? And now this is kind of like very counterintuitive, right? You're in the fasting state, right? You do not have like any exogenous source to make ATP. So you actually first you're gonna have to maintain glucose levels, and you also have to supply, to make this glucose, in these cells right here, right in the liver or whatever, right?
Mm-hmm.
So you need to also have ATP. Kind of counterintuitive, right?
Yeah.
Like where do you have ATP coming from in these cells? It's really important. So again, so once you start doing gluconeogenesis, the foremost, like this is number 2, 3. I mean, these are kind of like sorted together. The most single important thing is in gluconeogenesis you need is fatty acids breakdown. Because this fatty acids breakdown is going to give you acetyl-CoA, and it's going to give you ATP. So while you actually have this gluconeogenesis, you're also starting what? Little bit what? What is this blue a yellow line? This is, we'll just use this color. This yellow line is fatty acids breakdown. Right? Because if you're doing gluconeogenesis, you need fatty acids, because this ATP right here, this ATP, and this acetyl-CoA here, that is going to come from beta oxidation of fatty acids. Because every single time so let's say this is 16 carbons, right?
Mm-hmm.
16 carbons, and then it will become 14, then it will become 12, and then it will become 10, right? So you're breaking down every single time, 2 at a time, right? And this is every 2 at a time is giving you what? Acetyl-CoA. So, and every single time you're breaking down, you're getting acetyl-CoA. And what else are you getting? And ATP. So you get your ATP, you got your acetyl-CoA. Very good, right?
Wow.
So then you got your acetyl-CoA. I mean, it's much more complex than this, but we're just making it for simplification's sake, right?
Right.
So acetyl-CoA, you got your ATP, and then so you got your acetyl-CoA. This will activate your pyruvate kinase. You got your substrate skeletal muscle breakdown coming in and feeding in the pyruvates. You got your ATP, and now you're making the glucose. All this work for what? For making sure that RBCs are fed. Right, so this is so important, right? That's why we just go in anytime we see like 40 glucose levels and 60 glucose levels, we start shooting them D50.
Yeah.
Because it's so important, right? So now like your fatty acids are also going up, right? So now the question is what? Like at some point, so let's say you're doing gluconeogenesis, you're still not eating, right? And now you're kind of doing this process, gluconeogenesis, and it just plateaus at some point, right? It plateaus at some point. So the question now is, so I'll just actually color this fatty acids right here. So fatty acids also breakdown is going up too, right? Kind of simultaneously, right? Because it needs to maintain this, acetyl-CoA to keep this gluconeogenesis going on, right? So now what happens is at some point, let's say this is like 2-3 hours, at like 6 to 8 hours here, right? You still didn't eat, right? You're still doing your note, you still have some admissions going on, right? And you still didn't eat, and you're like, man, the cafeteria is closed, right? Which is always
Always
in our
the case.
always the case, right? And then resident lounge is out of the lunch, right? Except the day one of residency, right?
Yeah.
After that, it's always out of the lunch. So what happens now is that your body is like, well, if I keep on burning down these, at some point, do you want to keep continuing gluconeogenesis, right? So now your body at this point in time, some of this acetyl-CoA is shifted towards ketones too. Because what do you need to make ketones? You need acetyl-CoA. So some of this acetyl-CoA is start shifting towards ketones, right? So now what happens is at this point, you still have glucose right here, right? So at this point, prior to this, right, you still have glucose greater than ketones. So what did I say? The cells, if you are given pasta versus just a piece of bread, which one do you want?
I want the pasta, but
Right,
I'll
so
take bread.
which is glucose, right? So you will love glucose. Your cells love glucose, right? So every cell prefers glucose when the concentration of glucose in the plasma is higher than ketones.
Mm-hmm.
But what happens is if you keep on doing gluconeogenesis, what will you do? So you have to, number third is called have to. So we kind of already sort of started here is beta oxidation too, right?
Mm-hmm.
Oxidation of fatty acid, right? Right. So we kind of start doing oxidation of fatty acid. But can you just keep continuing gluconeogenesis all the time, at the highest level?
No.
No, right? Because what will happen is you will need to spare the glucose only for RBC. Because now you're making glucose for every single cell in the body, right? They're eating glucose, right? So now the thing is can you stop this gluconeogenesis altogether? You cannot, right? Because your RBC will not have glucose. They're the only cell. So what you can do is, how do you spare this glucose? So another way they say is, this is called synonymously, let me see. It's called sparing the proteins, right? Your body does not want to break down all your proteins, right? Your body's like, hey, it's already 8 hours. You still didn't eat. I don't want to break down your skeletal muscle all the way down, right? Because if I break your own skeletal muscle all the way down, you will not be able to move evolutionary from one point to another, right? Because of your muscle and the bones, right? So that's not sustainable. So what your body does is it spares the protein. Because why you spare the proteins? Because if in order to do gluconeogenesis, you need a lot of protein breakdown, right? So your body wants to spare the protein breakdown. So what does it want to do? Now it wants to start making more ketones than glucose, right? So what happens is a lot of this beta oxidation now you know, this acetyl-CoA will, even like this fatty acids breakdown, will go crazy up. And then gluconeogenesis will go down. Makes sense?
Mm-hmm.
So gluconeogenesis, this is the gluconeogenesis. So you can you stop the gluconeogenesis all the way to 0? No, right? Because you need to still maintain glucose levels, right? The glucose levels now kind of like going down towards 80-ish. Right? Not like exact 100, but
Yeah.
now you're because why? Because you're still doing gluconeogenesis right here, right? Here.
Yeah.
Makes sense? And this is a fatty acid breakdown. When the fatty acid breakdown going up crazy, what are you doing? You're increasing the amount of acetyl-CoA. Right? If you have a high amount of acetyl-CoA, you will make more ketones. Right? So now you have ketones greater than glucose. And now all cells use ketones
Except.
except RBCs and a little bit of kidney. We won't really get into this, but like mostly RBC. Okay? So we will just kind of like remove this. We'll just focus on RBCs, right? So RBC is getting this glucose, that's fine. And now you keep on doing ketones, ketogenesis, and you also doing. So this is what's happening, right? This is every day happening. And now what happens is, as soon as and then if you do not eat, you will burn down all of your fat. So once your fat runs out, you cannot do then this gluconeogenesis start going up, and eventually you will run through your old fat, right?
Mm-hmm.
And this is the kind of like science behind keto,
Keto diet.
the keto diet too,
Yeah.
right? Because keto diet, what they do is they don't feed them carbohydrate, right?
Mm-hmm.
So what they do is they actually give them a lot more fats, right? They're eating, so they start burning down their fat. They want to stay in this ketogenesis state, because what happens is a lot of people who are doing keto diet, what they do is they do not fast at least 6 hours, because they keep on eating, right? If they keep on eating, they will just keep on going into fat state. So keto diet only works if you also fast. You have to fast at least more than 8 hours to start burning down your fat at the highest rate right here, right?
Oh, I see.
You know what I'm saying? If you don't, well, you're burning down your skeletal muscle. So these people with keto diet, if they're just fasting only 6-8 hours, they're burning their skeletal muscles way much more than their fat. That's why they get a lot weaker. That's why people who do keto diet, they should actually practice, but again, I'm not expert in that, but that's the biochemistry, I think, behind it. But again, yeah. So that's the normal things that are happening in our body, right? So again, so what we talked about today is, so this is all to understand DKA and HHS and diabetes, right? Okay. So first, this is a normal physiology, right? So what happens during a feeding state, you want to maintain your 100 grams of glucose. When you eat, your glucose levels going crazy up, right? So how does your body interact that? It's by hormones, right? So you have insulin and glucagon. So in feeding pathways, you have way much more insulin, and how you remove these, excess amount of glucose from your plasma is glycolysis. First, you need to remove this glucose into these cells, right? Mm-hmm. So you need transporters on these, heart, skeletal muscle, and adipose cells. So these transporters are called GLUT4, that are upregulated by, that are actually translocated by insulin. So you have insulin, now this glucose start going into these cells, right? And then also in the liver don't have GLUT4 transporters, but what do they have is glucokinase, these enzymes that will trap the glucose, in the cells right here, right? In the cells right here. So this glucokinase is only upregulated by which hormone again? Insulin, right? Mm-hmm. So when you have feeding, you have high insulin, your transporters here, then you have in the liver, you have a glucokinase that will be upregulated, and now you're actually start removing this, glucose. So now in these cells, you will start burning down glucose, right? So you burn down to make pyruvate, right? So that's actually, and also you can take some of these glucose and shift towards glycogen. That's number two. You can store. That's allowing you to remove this glucose and then also keeping it in the cells, right? And also number third, you can also fill up your stores because you still want to burn. You want to make sure this glucose is back to 100, right? So you can actually do ATP synthesis, right? So you can actually store your creatine, phosphate. All these people, remember, like when they go to gym, they just eat like potatoes and stuff to get like charged up or something.
Right.
That is what they're doing right here. When they eat right here, right, they're actually storing their creatine, phosphate stores and ATP and all that stuff, right? Because again, for muscle, when somebody's going to work out, right, they need their ATP to muscle relaxation and contraction. Remember that ATP, myosin, actin.
Yeah.
So all of that. So then some of this will be shifted out, and you will make acetyl-CoA, and acetyl-CoA will be used to make fatty acid synthesis, which is this, and then some of this will be used to make cholesterol, and then it will be packed into VLDLs, sent out in the plasma, and then you have lipoprotein lipases on the top of the adipose cells that will take off some of these VLDLs, start cleaning them, and you start storing the cholesterols here, and then VLDLs will become LDLs, and LDLs are going to be higher, and VLDLs also going to be higher. So when you cleave these VLDLs, you will make something called IDL, intermediate DL,
Right.
and then you will make low density LDL, and then these will be higher. And that's why you do not want to measure these plasma levels if you're screening for lipid or hyperlipidemia. You want to make sure they're at least fasting, because if they're in the feeding, they just ate, they will have falsely elevated because you just synthesize a lot of these VLDLs, right? So that's what happens. So that's how you maintain your plasma glucose levels in 100, right? That's a normal physiology, and that's all. Its most important thing is your hold on, why did I erase that? Is insulin, right? This is really important because like in diabetic people, if you don't have insulin, none of this is going to work, right? So what happens in when you actually go into fasting state, right? Your body wants to maintain glucose levels. So what are the rules in fasting levels? Again, all cells prefers glucose more than ketones, and then all cells can use all cells can use ketones except one cell which don't have mitochondria, which is RBC. So that's why it's so important to maintain your plasma glucose levels because of the RBCs, because you want to keep your RBC working, because if the RBC is not going to work, you're not going to be able to maintain if your RBC is not going to work, right? So if you don't have 100 glucose is not present in the plasma, these cells will not uptake glucose because there is nothing in the first place to present, right? And then they will not be able to make ATP. If you're not able to make ATP, well, you cannot maintain your the sodium potassium pumps. Well, if you cannot maintain your sodium potassium pumps, the gradients equalize. Anytime the cell's gradients equalizes, the cell dies, right? That's why we need to maintain those gradients, right? So for that reason, these sodium potassium pumps pump the K in is working by ATP. Well, if you don't have ATP, which means you don't have a glucose, well, you will not this cell will lyse, right? So that's really important to maintain this glucose. And so how do we do that? So there's mostly like 3 or 4 pathways mostly important. So first, when you go into fasting, you're not eating at least for 30 to 60 minutes, your body goes into fasting. The first thing you burn down is glycogen. So basically glycogen is trapped glucose branches of on the glycogen molecule. There's a bunch of glucose attached to it. So now you're actually burning down the glycogen from the liver, right? Not in the skeletal muscle, only in the liver. We will talk about what happens to skeletal muscle and the glycogen, but mostly in the liver. Why? Because liver is the only organ that actually has glucagon receptors, not the skeletal muscle, right? So this we'll talk about later. So here you have which hormone is higher? Glucagon, right? So in the fasting you have a glucagon higher, and then which is lower? Insulin, right? So now the ratio of glucagon to insulin is way much more higher. And then what happens next is that you're going to start breaking down the glycogen from the liver, and that's helping you maintain simultaneously the levels of glucose right here, right? So you're maintaining that right here, right? So what happens is once you run out of, if you're still not eating 2-3 hours, right, you run out of your liver glycogen already, right? So the thing is you still need to maintain these glucose levels, right? Because what happens if you don't? This will tank down, and then RBC will not have any source, and again the RBC will lysis, and then this will, because they don't have mitochondria, right? So for that reason, you need to maintain this glucose levels, right? So what do? Well, you need to make the glucose from scratch, gluconeogenesis, right? Making glucose from scratch. So in order to make glucose from scratch, you need four things, right? Protein breakdown. You need a protein breakdown. Why? Because now you're actually using pyruvates to make what? Glucose, right? So these pyruvates, these oxaloacetates come from the protein breakdown, which is amino acids. Some amino acids are ketogenic, some amino acids are glucogenic, and some are both gluco and ketogenic. So you need mostly glucogenic or glucoketogenic. So this will give you what? These will give you pyruvates. In order to use pyruvate, you need pyruvate kinase. This will actually go backward, this reaction.
Mm-hmm.
So in order to upregulate pyruvate kinase, you need what?
Acetyl-CoA.
And you also need ATP, because remember, last time when you actually break down the glucose, you actually did what? You actually, once this is actually happening in the cells, you actually made 200 ATP.
Mm-hmm.
But if your liver, this is the most important organ, right? That actually doing the gluconeogenesis, right? So if you are actually making glucose, you need to start, breaking down this, fatty acids to actually supply you ATP, and then fatty acids breakdown also give you what? Acetyl-CoA, right? And then if you have ATP and now you have acetyl-CoA, now you can jumpstart this process, and now you're making your glucose. Now, really important question here is, remember there's in med school first year we studied an enzyme called LCAD and MCAD. Remember those enzymes? MCAD was long chain fatty acids breakdown, and the medium chain fatty acids breakdown. So if some of these people, they actually have deficiency in these, right? Mm-hmm. So if the kid is coming in, like maybe like 3 or 4 years old, right? And they're always becoming hypoglycemic in the fasting stage after 2-3 hours, right? Mm-hmm. So they if they have they're becoming hypoglycemic, you always want to be thinking about this, right? Because if you don't have this, will you have acetyl-CoA?
No.
If you don't have acetyl-CoA, will you be able to stimulate this pyruvate kinase? No, right? So even if you have breaking down the glucogenic and all that stuff, would you be able to push it backward here? No, right? So if you don't have MCAD, you won't have acetyl-CoA, you won't push this process backward, and these people will have low glucose in their plasma. Mm-hmm. They will become hypoglycemic, right? And also, if you don't have acetyl-CoA, will you have ketones? No, right? So people who have MCAD deficiency, so clinical correlation here is clinical correlation. If you have MCAD deficiencies, right? If somebody's coming in, they are hypoglycemic, their blood glucose is low, hypoglycemic, and they're hypoketonic, right? Which means they have MCAD deficiency. Mostly when we check people with a DKA, right, they usually have hyperglycemia and hypoketones, right? If somebody's don't have something like that, hypoglycemic and hypoketones, which means they're not breaking down their fatty acids, right? So you should always be checking for mutation. We'll call MCAD, right? So stuff like that. So anyhow, so that's the gluconeogenesis. So you're making this, so you're maintaining after you run out of your glycogen, you are actually kind of slowly starting your gluconeogenesis, right? At some point, after 2-3 hours, your gluconeogenesis is taking over. And also, in order to do gluconeogenesis, you need acetyl-CoA, right? Mm-hmm. This acetyl-CoA is going to come from what? Fatty acids breakdown, right? Yeah. This fatty acid breakdown is also simultaneously starting, but it's not crazy, going up, but gluconeogenesis is definitely going up, right? And now the cells are preferring glucose over ketones, but now you cannot keep breaking down your proteins, right? Yeah. So what do you do? Well, you want to spare the proteins, right? Your body wants to spare the proteins. How does it do it? Well, it needs to start breaking down fats way much more higher, right? But you cannot entirely shut down your gluconeogenesis, because if you do, you will not be able to maintain this glucose levels, right? Mm-hmm. So what happens is, let me just say here is, so now that your fatty acids breakdown is going up, right? So now, at some point, now I will just draw a curve here, somewhere here. Let me just change the color, blue color. So now there was some ketones here, right? But here the ketones start going up, right? So there's ketones starting trending up, going up, going up, going up, and now all of a sudden the ketones taking over, mm-hmm. right? Now these are your ketones here, right? So now your ketones is way much greater than your glucose, right? So now your body's is actually ketones is more than your glucose, and then your body is start using what? Ketones, right? So all the cells in your body, including neurons and everything, right, they start using ketones way much more because the concentration is higher. But your RBC cannot use ketones because it doesn't have mitochondria, so it still has to use glucose, right? So that's how your body is like sparing your excessive amount of breakdown, protein breakdown. Okay. So this is like normal fasting and feeding. So now we will go into DKA.
All right.
Okay. So the next chapter we'll talk about like part two of this series will be what happens in diabetic people, DM1 or DM2, right? Diabetes type 2, type 1, right? So we'll talk about what happens when you don't have insulin, right? So again, these pathways are maintained by insulin, right? So what happens when you don't have insulin? What is happening? How does your body is fighting? So in order to understand this, like, in order to understand DKA, you need to understand these things first. If you understand these things, what's happening, so from next time, I want you to, anytime you eat, I want you to think about these things. Anytime you're not eating, I want you to think about these things, right?
Yeah.
So, and then that will give you better reference, like, especially like, if you're working out too. So like, that, you need to go eat, because otherwise you cannot keep breaking down. And that's why a lot of people like, first, if they're excessively working, not eating, they will start losing weight excessively,
Yeah.
right? And that's what happens, right? And that's why, like, that's the like concept behind keto diet, is you do not eat, you're fasting all this time. Because if you're fasting all this time, you're going to spare your proteins at some point after 8 hours, and you're going to keep on breaking down the fats.
Yeah.
And that's the whole point. Okay. So I think that's where we should, end